All right. I'm kind of in a funk right now, so I'm not exactly high energy. So we're just going to kind of do it to it. And we did not eat dinner, so I'm still kind of snacking. Give a minute for a few of y'all to get on. I know most of y'all will just watch the recorded video. Turn off the vent in there. Give people another minute. All right. Y'all can go ahead and start like asking your questions or whatever, and I'll answer them as we get going. Just writing stuff down so I have it ready to go. Okay, can you go over hydroponic and hypertonic? Scarlett, I know what you mean. I know you mean hypertonic and hypotonic. Hydroponic is a way we can grow plants. And one way, very popular right now, is not school appropriate. So I know you mean hypotonic. Okay. What do you use may active transport? No, it's ADP, but like there's a protein. Does it something? Oh, wait. Ooh. Okay. Berg, I'm glad you mentioned. Oh man, that was kind of rough to read. All right. Yeah, I'll look at that. Okay. Let me take one more bite of food. All right. Okay, so Scarlett and Burke, I'll go ahead and look at your questions right now, and then we'll back up a little bit. All right, so they're asking about transport and osmosis, water movement, that kind of thing. All right. So we did this in class. There are two main kinds of transport in a plant. And I feel like my light is off or something. Like you can't see very well. All right. Two main types of transport. These are not the only type, just the main types. This is how molecules get in and out of the cell. Some of them are passively able to go through, which means no energy. Literally the cell does not have to think about this or do anything with it. It's going to happen naturally on its own. If it's passive transport, it'll be things like diffusion. Diffusion means, if this is our cell membrane, diffusion means it can literally go through one way or the other without any kind of problem. All right. The only thing that can diffuse that way without without any kind of help of a protein or anything is gas, such as CO2 and oxygen, right? The other kind of passive transport would be for water, and we call that transport osmosis. So again, water is going to move from one side to the other. Facilitated diffusion, again, there's the word diffusion, so you know it's passive. Facilitated means it's got to use some kind of protein. So now we've got our membrane, but within our membrane, I feel like this orange isn't showing up very well. Let me see if I have a different color. In facilitated, we have to utilize a protein to help. Okay, that protein is just helping to protect molecules from these fatty acid tails that are all in here. Okay, because things are not able to come in contact with those fatty acid tails if they're polar. So this protein creates the tunnel to allow things to slip in and out. Things that might need help with this are charged ions or things that are large, such as glucose. But still, here's the ultimate driving force of a passive transport. It's what kind of concentration the molecules are going from and the concentration the molecules are going to. Passive transport. Remember, let me kind of do it on this one. Passive transport is the movement from a high to a low concentration. I'm going to kind of just draw. You can't really see dots too well, so I'm just going to do this. Okay. Passive transport is high to low. Molecules do not want to be concentrated, so they move to where there's less of them. This is able to happen naturally in your cell. That's why no energy is needed. So water, there's a lot of water, it's going to move. Why is it going to move? Because there's actually a lot of salt on this side, and it's going to equalize, bring things to an equilibrium. There's a lot of glucose on this side. It wants to move, okay, from a high to a low, so it's going down its gradient. In an active transport, we end up having to use ATP energy because now it's the opposite. We're moving from a low to a high. So we're forcing molecules where they don't want to naturally be. Nobody wants to be concentrated. Molecules are the same. They want to move where they can. All right. So in an active transport, we're going to move things like sodium and potassium ions. Na plus and K plus. We're going to move these ions, right, against their gradient. This is actually nerve impulses. Why you can feel things, why you can see, why everything works in your body. Your nervous system is moving those ions against their gradient. So what you get, I'm afraid my dark pen. Go ask Daddy for a darker pen. Because I don't think I have one. Hold on, my pen's kind of running out. Hold on while I'm doing this. Scarlett, we're going to get to your hydroponic. Not hydroponic, but your, let me get to your question. Burke, I'm explaining. Grant, I'm about to explain your phagocytosis question. Osmosis doesn't make sense. Like, how do you know if it's hypo or hydro? There's no such word as hydro. Like, hydro is water. We're talking about hyper and hypo. Get those words right. I have a pink one. Okay. All right. So, active transport. Let me get this going. Okay. In an active transport, the molecules are concentrated on one side and not so much on the other side. So what we're talking about is one side of the cell versus the other side. This is a membrane. In an active transport, molecules are going to be forced to go back to their his pen's almost out too. They're going to be forced to go back to their concentrated side. They don't want to, so they get forced to do it by energy ATP. Active transport comes in a couple of forms. So sodium, potassium gets forced. That's your nervous system. Or we can engulf things or push things out. And we call that endocytosis and exocytosis. Endo means into, and cyte means cell. Those are prefix suffix words you just need to know. And then exo is out of, and again, cyto for cell. The difference between them is, are we bringing stuff in or are we pushing stuff out? Again, actively. So endocytosis has two ways to bring things in. A cell, just like, think about you as like if you're one big fat cell. How do you take in molecules? You can eat it or you can drink it. A cell can eat or drink. Phago means to eat, pino means to drink. Your examples are, in a phagocytosis, that's a white blood cell eating a bacteria that then will get all kinds of chopped up into little pieces, and then we'll exo it. We'll push it out of the cell so it becomes waste. Okay, endo and exo are two types of active transport. Phago and pino are just two ways that endo can occur, because a cell can either eat it or drink it in, endo for in. So it's the difference of phago or pino. So, right, I'm going to kind of draw this. Okay, so imagine this. You've got an immune system cell, right? Looks real hungry, right? You've got a bacteria. Ah! It's screaming because this immune system cell is about to eat it. And when it eats it, we call that phago. Now, I would love to, draw it, where he's all like, yum, right? And he's like gobbling it, right? It's not really what happens. What actually happens is the cell gets close to the bacteria and forms around it. So he's like, so this immune system cell will actually eat that bacteria and take it inside and make a little vesicle for it. Makes a little sac. All right. This would be phago. The only difference, the only reason pino looks different is this isn't a cell. That's just dissolved something in a liquid. So it may be sugar, maybe salt, maybe hormones, but it takes it in by drinking it instead because it's not an actual cell. All right. So phago, eat, pino, drink. Those are prefix suffix terms you need to study. All right. All right. So Burke. So let's just kind of go back. Burke, your question was, what is used to make active transport? I know it's ATP. Yes, it is. But like there's a protein does it or something, I don't know. So in active transport, a protein is still there. Okay, a protein is still helping. So let me kind of show you these two things. Okay. All right. So 2 cell membranes, protein. The first one I'm going to show is passive. So this is like glucose. Whee! This glucose molecule is going through the protein from a high to a low. Right? This is passive. But how this looks different here would be glucose going from low to high. Whee! But then something is there. Okay. The glucose is now going from a low to a high. It's being forced. Bam, ATP is there. ATP is present. So anytime you see ATP, you know it's active energy. active transport. But then, yes, a protein is still utilized because it's got to move these molecules against their gradient. So yes, Burke, both are technically there, okay? It's just going from a low to a high or a high to a low. So the second one is active transport because ATP is there and you can see the direction it's going. All right, phagocytosis, I already answered that for you, Grant. It's the eating. Osmosis doesn't make sense. How do you know if it's hypo or high? I'll do that again. Hypo is when there's less of something in the cell. Okay, here, let's kind of make this distinction, Toby, because I'm pretty sure, Toby, you're Jurassic Parkour, if I remember. Okay, guys, the terms hyper and hypo can refer to a solution outside of the cell and a solution into the cell. So do not get used to saying that a hypo is always the cell or hyper is always, no. You label the solutions first. Who cares where they are? We don't care where they are at first. We will care at the end when we're trying to describe what happens to the cell because of where the water was and how it's moving. But first, you just compare their solutions and label them hyper because it has more, hypo because it has less. And we'll practice that. Okay? All right, hold on. There's many questions. Let me go through it. Yeah, what do mean? What that mean? Okay. Okay, so Scarlett, you're asking what's a gradient. Guys, the word gradient literally is the difference between two places. Your driveway has a gradient. It's a higher up here, like closer to your garage, and then it slopes down as you go into the street. That's a gradient. When I draw these pictures and there's more dots on one side than there is on the other side, that's a gradient, right? I can represent this as well by drawing what I did in class. Let me do this on the other page. Hold on. In class where I draw the, just a slope showing a high to a low. There's a lot of something up here and there's not a lot of something right there. That's a gradient. So if I were to draw this like a picture of a beaker or something, and I drew a membrane that's semi-permeable, like it can go from one side to the other. A gradient is because one side has more of something than the other side does. So I've created a gradient, a high gradient, a low gradient. A lot of solute, not a lot of solute. Hyper, hypo. Right? Shouldn't those be different? Shouldn't those be different? I don't know what you're referring to. But doesn't hyper mean over? Hyper does mean over or above. Hyper is not less. Hyper is more. Hypo is low. Just think of it. Hypo is low. Right?
Low salt's not water.
Low salt, not water. Yeah. Because the word tonic refers to the strength. Is that you blowing up or me? Oh, Kylie's text. Okay. Kylie, I'll do organelles and stuff as well. And I'm going through the questions right now. Semi-permeable, the word permeate is when something can pass. So if it's semi, only some things can pass. The cell membrane is just that. Only certain things can pass. Guys, that was your diffusion lab. Starch stayed in the bag. It did not leave the bag. So it was not permeable to it. But the glucose did leave the bag. That's why it tested positive in the cup. Because that dialysis tubing we used was semi-permeable. All right. Hypo is low. Yes, I'm probably wrong. Thanks, Scarlett. Okay. Nom, you are born to wean. Okay. How do you know? Yeah, I'm Toby. Okay. It's null. Okay. So ATP forces the glucose backwards on the protein bridge. Okay, yeah, so protein, yeah. So good way to say it, yes, a protein bridge. ATP if it's active transport, will push things from a low side to a high side of that molecule. So again, gonna kind of just keep showing this so maybe we can feel better about it. All right. So I'm just gonna actually draw it like an actual cell. I'll draw a cell, and we're gonna put some solute in that cell. Doesn't matter what solute it is, it's just gonna be a solute. Okay. So here's a cell. I'm gonna make a gradient. Let's say there's 50% of a glucose molecule inside of the cell. And there's a 10% of the same glucose molecule. So this is going to lead into osmosis here in a second as well. So this 10% glucose versus 50% glucose. If we talk in the word of a gradient, where's the high gradient, right? And the low gradient. If this was passive transport, it would go from a high to a low. So glucose would move, okay? But in an active transport, we're going to force glucose where it doesn't already want to be because it's already crowded. So in an active, we're going to force it in, the glucose, right? Transport is for moving of the same molecule. Now, I'm going to use this exact same picture, and hopefully this makes you go, oh my gosh, you got to be kidding me. That was it? So I'm going to use the same picture of glucose. But now also let me go ahead and write the water out as well, okay? So if there's 50% glucose, that means how much water?
50.
50% water. Okay. If there's 10% glucose, how much water?
50.
90% water. Right? Always out of 100. All right. So using the same idea. Passive transport is going down their gradient, going from a high to a low. Active transport is forcing it where it doesn't want to go. So in this situation, if it's permeable to molecules, then they're going to move where they naturally want to be, which is not concentrated. So glucose would want to move out. Okay, it does not want to be in here. But let's say in this situation, the semi-permeable membrane, glucose can't move. Let's say glucose can't, but water can. And water is desperate to do what? Make things equal or homeostatic, right? Your homeostasis. So look at those same numbers just like we always would. Okay, so let's say we can't move glucose. So ignore the glucose for just a second. Now compare the water. Compare those numbers of water. Where will water want to move? Like just not even thinking of glucose, not thinking anything like that, guys. Nothing like that. Just compare water. 90 to 50. Where would water naturally want to move? Not if it's 90 versus 50. So water is outside the cell. It doesn't want to be there, right? Because there's too much of it. There's 90% of it. So it wants to go where there's less of it, which means in the cell. And now bring that glucose back in and look at what's happened. 10% glucose was out, 50% glucose was in. Water went where there was more solute. But water also followed the same concept of passive transport, guys, from a high concentration of itself to a very low concentration of itself. That's how this whole world of osmosis works. Because, guys, osmosis is passive transport, high to low. Water moved from high to low, which brings us into hypertonic, hypotonic, right? So let's label that. Compare the 50 to 10 of glucose. Now remember, because the hyper and hypotonic is the strength of the solute. So we need to look at glucose. So 10 to 50 Just compare the numbers. Whichever one's greater gets the word hyper. So that means inside my cell, I'm going to write the word hyper, and outside my cell, I'm going to write the word hypo. And it still works. Water will always go towards the hypertonic solution, right? So when we do these osmosis problems, it is a passive transport because molecules of water are moving down their gradient from high to low. They're moving where there's more salt and less of them. So they're like, sweet, I don't have to be all crammed up with myself, and I can go ahead and make things equal. So I'm going to mosey on over here to this side of the membrane. Okay, I'm going to give you a couple more of those. All right, so Nico, in the active world, let's say glucose can move, but with the help of energy, okay? So if glucose can move with the help of energy using, like you phrased it, a protein bridge. So there would be a protein. And there's proteins embedded in the membrane. And if it's active transport, then it's going to go where it doesn't want to go. So that means this 10% of glucose would come in against the gradient. So it went from a low to a high. And that would be an active because it's got to use ATP to do that. Because glucose didn't want to do that. There was already enough glucose here. And again, it's the same on this picture. Wherever there is more of a high or low gradient, active forces it to the high side again. That's why ATP's got to be there. Or does the ATP let it pass, just forces it into a small place? No, ATP actually activates the protein to open, to allow glucose to come through. Without ATP activating that protein, glucose isn't going to go anywhere. All right.
It's like, nah, bro.
Why would it do that? Like for energy or power? Why would what do what?
James,
Why would glucose?
James said no.
Yes. Why would glucose want to have to come in? Again, this is your nervous system. So a nerve impulse. So like you step on a tack, right? And it hurts. You know, forget the tack. You stepped on a Lego piece, okay? That's like death, okay? So you stepped on a Lego piece. The instant you stepped on it, a nerve impulse went shooting from your foot up to your spinal cord, back down to your foot, and allowed you to feel it, reflex your muscle to pick up your foot and say, ow. That was only possible because of active transport, right? Constant movement of molecules in all systems of your body, the nervous system, the muscle system, all of that. So when you say, is it used for energy or power or whatever, it's used for all biological systems. Active transport is necessary because when we talk about water trying to maintain homeostasis, all of your molecules are also trying to maintain homeostasis. So some of them move one way or the other to maintain that. And sometimes to do that requires ATP energy. We're going to end up next unit looking at photosynthesis and respiration. ATP is needed.
Photosynthesis uses carbon dioxide.
ATP is needed in those, and you'll see how every time ATP is used, it forces molecules against where they want to go. So they get forced into a higher concentration. And because of that, plants can photosynthesize. Because of that, your cells respire and you get energy. Okay, active energy store up glucose. Glucose is just a one example that I use. It can be the movement of anything. In photosynthesis and respiration, when we look at it, that's actually hydrogen. So ATP and energy is moving hydrogen. So it's not, ATP's everywhere. Okay, it's just anytime you see ATP at a protein, it's activating that protein. And whatever it's moving in, it can be anything, guys. Whatever's being forced in or out, if ATP is there, it's telling you it's forcing it to a high concentration. away from a low where it doesn't want to be. So more solute is hyper and less is hypo. Yes. Y'all have to just remember hypo is low. Just remember that little rhyme. Hypo is low. All right. So let me give you a couple of pictures. So the number one thing, and I kind of stressed this in 7th period, is y'all have to not think about the word hyper and hypo in relation to a particular solution, like a cell is hypo. No, just look at the numbers, okay? Okay, if I just gave you those numbers, if I gave you these numbers and I told you both of those are the salt, okay, gave you the numbers, both are salt, just look at it, okay? If you were given these numbers, what would be labeled hyper because there's more or above, and what would be labeled hypo because it's low? And so once you've just compared those numbers, you've labeled. So I would now just write hyper, and I would write the word hypo. And then, is it the cell? Which of these solutions was found where? After you've labeled it, now go ahead and figure out, was it the cell that was hyper or the cell was hypo based on the problem that I gave you? And if the problem said, if a cell containing 12% solute concentration was placed in a solution of 75% solute concentration, right? So right there, now I've told you. Now I've actually told you what they are. But guys, I didn't tell you, but you still could label it. So the cell is 12. My outside solution is 75. But now you also can say, well, which direction should water go? Well, water always goes towards the hyper. Okay? Water goes where more salt is. So if water is going towards here, and I've already labeled this as my cell, that means water is going away from this and into this. So water is going to leave the cell, and think about having a water balloon and squeezing, just like it's still open, like imagine it, and just squeezing it out. Or imagine a sponge, right? You're squeezing it. Eventually, that cell's going to shrink because it doesn't have as much liquid in it. Okay, it's not as big as it was. But y'all have to first just label the percentages, hyper and hypo. All right, what does a gap junction do? Does it allow things to transfer between the two cells, or is it just for communication? All right, so first, you're in Martin's class. I don't go into gap junctions or cell marker proteins on the surface of the cells. That's something I would go into for my AP class. So your gap, I don't know what you mean. Does it allow things to transfer between two cells, or is it just for communication? A gap junction is actually an opening between two cells. So how it's phrased is kind of weird because communication is the movement of molecules, right? But really, a gap junction just means there's a tunnel between this cell and this cell, right? So it lets things go through. A gap junction is most notable in things like heart cells. Gap junctions are what allow the electric impulse to pass quickly between your heart so that your heart keeps pumping and all the cells are synchronized. So it is technically just an opening to allow things to move between two cells, but technically it's grouped under communication. But I think what you mean on that one, John, is when she's talking about cell marker proteins are used directly for communication between cells. Like, and again, my kids don't worry about this. This isn't you, so just hold on.
Oh, great.
All right, so you've got two cells, John. If these cells are close to each other, like muscle cells or epithelial cells, then they can have a gap junction between them. That gap junction allows something from one cell to get passed over here to this cell. All right. But then these two cells can communicate through marker proteins on the outside. And so a glycolipid or glycoprotein, the stuff that's on the outside of the cell called markers, think of them as the hands of a cell. So if this cell has these kind of glycolipids, right, my fingers, and this cell has this kind of glycolipid, they will communicate with each other directly. And if it's a good handshake, then that tells the cells, hey, you belong here, that you're good. But let's say, Gracie, are you eating ice cream? Give me your fingers. Give me your hand. Let's say this cell, which has these kind of fingers, those aren't mine, and my cell come in contact with each other.
Swollen?
My hand comes in contact with her glycolipid proteins, the surfer proteins. I'm going to be like, what the heck? Who are you?
Crazy.
That's their way to talk to each other. That's communication. And then what that ultimately does is it causes an immune response in my body, because this cell is like, who the heck are you? I don't recognize you. You don't belong in this body.
No, I just see actually one.
And now my immune system is going to attack this cell as a foreign body because its surface proteins aren't mine. And I kill it. It's dead, right?
Still doing my
Now, why would that even happen? Where'd my pin go? Transplants. People who get organ transplants. That's why they have to take anti-rejection medication, because the surface proteins on that liver that they just got are not their surface proteins. Mine are different than yours. They're different than my children's. And so that transplant has a unique surface protein. And so my body's going to look at and say, what? Attack! And we're going to kill that organ. That's why so many people do have to get replacement organs, because they eventually fail. And it's not so much that they fail at communication because of those surface proteins.
like marshmallows.
Okay, John, I hope it answered your question. What's the purpose of the rough ER proteins? Okay, the center nucleus has the ribosomes, right? Nucleolus, Panda Express, way to go. Yep. Okay, I agree. Okay, so let's look at some cell organelles.
Okay.
What?
I was going to see if you answered my question. Go ahead.
Okay, so when it comes to the cells and the organelles, you've got the chart. I'd recommend, just kind of doodling on that chart. But then, guys, y'all have got to draw it. I've stressed that. I don't care how you draw it. Woohoo! I just drew a circle. No, it's not. That's a cell membrane, buddy. What is it also? It's a phospholipid bilayer. If you draw it, okay? Okay, so you've got your cell membrane. Then I'm going to draw another circle inside of it. What did I just draw? The nucleus. I'm going to shade part of that nucleus in, and now I've got a nucleolus. I'm going to draw some squiggles away from the nucleus. That's the ER, but now I'm going to make a distinction between rough and smooth. The rough has dots, the smooth doesn't.
Well, this is where I have my question. Okay, so like the rough ER is closer to the nucleus, and that's farther away it goes, the smoother it gets.
And I'll tell you why in a second.
Okay.
All right, I'm going to draw some more squiggly lines. That's the Golgi. It's the furthest away from the nucleus because it's got to be next to the cell membrane. I'm going to draw mitochondria, right? Mitochondria has like a hot dog with mustard on it. You've got lysosomes, which are just these larger little circles everywhere. I'm going to turn this into a plant cell, and I've made myself a pretty cell wall. And now I'm going to turn myself into adding some chloroplasts, which looks like they have stacks of coins. All right, vacuoles all in here. All right, now if you can doodle something like this, quick and easy. Look, it took me, 20 seconds? Talk to yourself while you're doodling it. Name what they are. And now also go back and do the functions. Cell membrane lets things enter or leave the cell. The nucleus holds your chromosomes. The nucleolus, ribosomes. The rough ER can make proteins. Why? Because ribosomes. Why does the rough ER have ribosomes? Because it's right next to the nucleus. And what just came out of the nucleus? Ribosomes.
Out of the nucleolus.
So they will attach to the rough ER. The smooth ER does not have any because the smooth is like fat, because that's what the smooth ER does, is makes fat. The Golgi is closest to the cell membrane because it's going to package things and ship them out of the cell. So it's got to be close to the membrane so it can have quick access to get things out. It's the post office. The mitochondria is for energy. The chloroplast is photosynthetic energy. Lysosomes, think lysol for cleaning. Again, ribosomes, think ribs for meat, protein. The cell wall is for structure and support. And then you'll have a large vacuole if it's a plant cell for storage.
Don't we have
Okay,
vacuoles?
we have small vacuoles.
Or
You
not?
need to stop, dude.
Stop what?
I don't know.
You talking to me?
No.
Who is that?
Burke's telling you need to stop, dude. Like, what does that mean?
Wait, what? Who's
Gabe? Not you.
Read it out loud, Mom.
I know. Is that what I'm hearing?
Yeah.
All right. Bye, person. Okay. All right. Okay, that person's gone. Where else?
Who was that?
I don't know. I don't know. I see that they commented on something earlier, so I removed them. All right, they're gone. And if they come back, I'll just remove them again. See, I get to do that. Why? Because I'm a teacher. Okay.
Well, what if it's one of the people who are also on here, they're running double?
They probably are. Okay. All right. Kendall, I have a question. So going back to osmosis, can you go over diffusion stuff? I will. Okay. Also, again, don't forget microscope. Basic things are on there. Always skills like independent, dependent variables, graphs, that kind of thing. Does the Golgi decide the reception transport? The reception transport, not exactly sure how you're phrasing that. Like, does the Golgi, are you saying when the Golgi receives, does it decide where it's supposed to ship to? No, technically. When things are made, like proteins are made, they actually have a signal on them, a chemical, and that chemical actually tells them where it's supposed to go. And so just think about, like, you walk into the post office and you already have a shipping address ready to go. So the post office doesn't get to choose. They have to follow the shipping address. The molecule has that shipping address already on it. Okay.
Good now.
Oh, thank you. It's like I've been doing this for a while.
Really?
Okay. All right. So again, draw it and just talk it out. Okay. Basic function and just the structure. I can't do, I can do it. I can do this 50 million times all I want. I need you to do it. Can you jot a general picture and call things out for what they are as you do it? That's the only way you're going to get better at the cell organelles is by drawing it. Okay, again, the only way you're going to get better at hypo and hyper is to keep practicing it. And guys, on those questions, draw them out. All right, if I've worded it, draw it. So again, I'm not going to give you an actual cell. I'm just going to say, if this is a membrane, I'm not going to tell you which side of the membrane it is right now. If this is a membrane, and in the problem, in the math problem, blah, 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 blah. Okay, blah, 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 blah. Blah, 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 blah. So blah, 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 49% salt, blah, 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 30% salt, blah, blah, blah. I'm not telling you what they are, where they're found. I just want you to focus on the numbers. So let's put those numbers in a picture. So 49% versus 30%. And I told you that was salt. So right there, compare those two numbers. So 49 is greater, so 49 gets the label hyper. 30 gets the name hypo. Water always goes where? Towards the salt. Again, ways you can think of it. I don't care how you think of it. Water loves salt. So water goes where there's more of it, right? Salt sucks. So salt will suck the water towards it. Grace?
You know, like when you go to the beach and the waves come up and like the sand kind of has salt, so like the water's going on to the sand and stuff.
Okay, Gracie's saying, imagine this like a beach. Sand is like the salt, so water's always rushing up to the sand, so water's rushing up to the salt. Whatever you want to use. Okay, so if you know that water goes towards hyper, draw water going towards that side. Because water is always inside and outside of cells. So you just have to figure out, is the water inside the cell leaving, or is water outside of the cell coming in? You don't know yet what's what, but just draw an arrow going towards the hyper, because it will leave the hypo environment and go towards hyper. All right. So now if I go back to this and I say, okay, blah, 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 blah. What if I actually change that blah, 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 blah? And actually said a cell, 49%, 30% salt solution environment.
You just flipped all of them off.
Good. Okay. So now I've told you what they actually are, but you've already drawn everything out. You already now you just have to tell me what happens with the cell. So now I'm going to draw a cell around hyper and at 49%. Water is now coming into my cell because that picture. And just like you would fill up a balloon, a water balloon with water because water's going into it, at a certain point, it can only take so much and eventually it will burst. Okay? And so we call that lyse. Okay. All righty. Let's see. Nikolai makes the ribosome. Oh, nucleolus. It's called a nucleolus. Makes the ribosomes, which leaves the nucleus, then rather sticks to ER or floats around. Correct. Ribosomes can either attach to the rough ER or they can actually be floating around in the cytoplasm. It's either one. Proteins can be made at either. And again, why would a protein be made at a rough ER versus a cytoplasm ribosome? It comes down to the signal that they left with, or the signal that the DNA is telling, like shipper. So again, so imagine my package is protein, right? I am going to take it to the post office so it can be, delivered. But where it got like that package itself, I'm going to put the little shipping label on it and I'm going to tell it where it's supposed to go. Where I packaged that box can vary. I could have packaged that box at home. I could have packaged that box at school. Okay. So I can package and create my proteins wherever I need to, rough ER or cytoplasm. Okay. What is the function of the smooth ER compared to the rough? Smooth, think smooth like fat, because the smooth ER makes fat, makes lipids. The rough ER has ribosomes, therefore it makes proteins, okay, because ribosomes ribs. All right. Kendall, I have a question. So going back to osmosis, could you go over diffusion? Again, Kendall, all diffusion is a type of passive transport, which is the movement of molecules. Diffusion can describe anything, but most specifically, things that can diffuse in your cells is gas. CO2 and oxygen. Those can diffuse the easiest. But also in class, when I sprayed that perfume by my desk, but then eventually you all started smelling it, that was diffusion. Any molecule can diffuse. It's just when we're talking about our cells and our cells have a membrane around it, then only certain things can diffuse. And those certain things are gas. But in my classroom, there wasn't any kind of membrane it had to worry about, right? So it just diffused. That's why some of you in your house have diffusers, right? That little thing that's all cute your mom bought from Scentsy. and every once in a while it's puffing out. You know, you see the smoke coming up. It's because it's actually oils and water and it's diffusing it, and eventually you smell it in your living room because those molecules can go anywhere. And that's what they do. They go from a high to a low. Okay. All right. Okay, what else can I answer?
Miss Good.
Hmm.
I will not be in class tomorrow.
All right. Again, passive and active. I can't stress it enough. I need y'all to just focus on the numbers first. Don't focus on the cell versus the, just focus on the numbers, label it like I was just doing, and then show me how water would move. And then if water's leaving versus coming in, that's where y'all need to describe shrinking, lysis, that kind of thing. What do lysosomes do again? So when you see the word lysosome, I need you to think Lysol. Because Lysol cleans, that's what lysosomes do. They clean the cell. All right, so again, back to the hyper and hypo and stuff. So if a environment solution was hypo versus hyper. Okay, so like I did in class, if the environment surrounding a cell was hypo, that means the animal cell or plant cell is hyper. That means water is going to move in. Then an animal cell will lyse eventually. It will swell and eventually burst. But in a plant, we call that turgid because a plant cell can't burst. It's got the cell wall, so it puts pressure on it, which causes a plant to stand up. That's why when your plants look all wilted and dead, your mom tells you to go water them. Give it a few minutes and they will start to perk back up because they've become turgid, because turg means swollen. Vice versa, if the surrounding environment was hyper, which means these are hypo, then you're going to shrink if you're an animal cell, or you're going to plasmolyze if you're a plant cell. So just kind of show you what happens. If you're a plant cell, you become turgid. If it's a plant cell, it becomes turgid, which means it actually expands. The vacuole does. Puts pressure on the cell wall. It makes it stand. Or that same cell will shrink if we put it in salt water. Because now the cell, or not shrink, but plasmolyze, because now that cell membrane tears away from the cell wall. As that vacuole becomes less and less water, it has less and less water, and eventually it pulls the entire cell membrane in. We saw during the circular organelle, that little circle one, that one was the lysosome. Lysosomes will just be a large circle in a picture because that's all they are. And so lysosomes clean. So just like in my picture right here, these are lysosomes. All those big circles. A ribosomes would just be little dots. Okay? Little dots are ribosomes, circles are lysosomes. Are there going to be any questions over the people who contributed to finding cells? If so, can you please go over those? Yeah, scientists are on them. You only had to know, what, 4, 5? And
We only had to know 4 or 5.
you only have to know their name and their general, what they did. So, and in order, based on discovery, you've got Hook, you've got Leeuwenhoek,
That's not confusing.
you've got, why?
Look,
Why is,
Leeuwenhoek.
well, if you pay attention in just a second, it will all help. Okay, so here are your scientists. In order of discovery. And oh my gosh, are they also alphabetical?
Wait, actually?
Yeah, they are.
That's helpful.
Hook, Leeuwenhoek, Schleiden, Schwann, Virchow. Okay, so alphabetically in order, Hook discovered the cell. Leeuwenhoek discovered animalcules.
Animals.
First one to use the compound microscope. Schleiden, plants. Schwann, animal cells. Virchow, pre-existing. And then Virchow, cell theory. So they all kind of built up on top of each other. So Hook, look, I found this thing. I'm going to call it a cell. Look, guys, I found all these tiny things that actually look like they move. They look like a cell, but they move. So I think they're animals. They're just really small. Schleiden, hey, did you know plants are made up of cells? No way, Schwann. Yeah, the animal cells are made up of animals are made up of cells too. No way. Yeah. Virchow comes in. Hey guys, thanks for your work. I'm thinking if animal cells grow, plant cells grow, or plants in general grow, then that probably means a cell has to create more cells. Oh, something called a pre-existing cell. Thanks, guys. I'm going to help create this thing called a cell theory, made up of 3 things. Basic unit of life. Thank you, Hook. All living things are made up of cells, Schleiden and Schwann. And cells must come from pre-existing cells, Virchow himself. Okay? When water is leaving the cell and the membrane tears away, since the water is escaping, would it be passive and osmosis? Well, osmosis is passive. Like osmosis is passive transport. It's the movement of water. So, yes.
I osmosisize.
Okay.
I've got a question.
What?
Will there be a bonus question? If so, can I write it?
No.
Please?
No.
It's going to be true or it's going to be what is the color red? True or false?
You do hear her.
You have a 50-50 chance of getting it right.
Oh my God. Are we
but
done?
don't tell me which one it is so that I can guess too.
Are we done? I think we're done.
No.
Yes, I vote yes. Nico says yes. I vote yes.
Yes.
Gracie, what? Yep. All right. Rewatch again. Things. So one task sheet. Look at that. I would vote true. Task sheet. Be sure you take care of that. Practice. Look at some of the practice stuff. The osmosis problems. Go, look through the osmosis. On your task sheet, that osmosis worksheet's on there, so y'all could practice that again if you still need some examples. And you can see me. I won't get there till probably at 8 tomorrow, just because we don't have to be there early. So, but if you get there and you want to swing by at 8 and ask the last minute questions, that's fine. Here's the thing. If you come in, first thing I'm going to do is I'm going to ask you to do the same things I've been asking you to do already. Can you draw a basic cell and label its organelles? Do you know how to do that? Second thing, hyper versus hypo. Can you identify between the 2 numbers? And then draw the arrow which direction it should go. If you can't do those things, you better be practicing right now. All right?
I won't be there for your class either.
Yeah, Burke, then you'll just have to either come in during Mega. Actually, you can't come in during Mega because I'm not there tomorrow. So you'll just have to take care of it. What's tomorrow? Wednesday? Thursday before school.
Yes.
Or Thursday during Mega. Okay? All right, guys. All right. Watch the video again if you need help. All right. See me at 8 a.m. tomorrow. Bye.
They want the question. You see that? They want the true or false question.